and welcome to the lecture 2 of the series of reproductive biology. We have understood about how the steroid hormones get synthesized in the cell from the cholesterol, which is 27 carbon compound. Now in today's lecture we will see how the important hormones of reproduction, how do they perform their action. So here I have enlisted a few hormones which play significant role in reproduction, which includes estradiol, testosterone, progesterone, and then there are GnRH, FSH and LH. Now estradiol, testosterone and progesterone, you have just learned in the first lecture that all these three hormones are steroidal in nature, because of which they are able to cross the plasma membrane and hence they do have receptors inside the cell. So they do have cytosolic receptors, they have nuclear receptors, and recent research have shown that they also have receptors on the membrane. But if we look at GnRH, FSH, LH, they are glycoprotein in nature and they cannot cross the cell membrane. Hence, they have a membrane bound receptor which is G-protein coupled receptor, that is GPCR. So, we will first start understanding the mechanism of action of the steroid hormones. Before explaining you their mechanism of action, I think it is of utmost importance that you should know the structure of the nuclear steroid receptors. So, steroid receptors, they basically belong to a super family wherein there are receptors for androgens, estrogens, progesterones, then thyroid hormones, vitamin D, retinoic acid, nerve growth factor 1b, steroidogenic factor 1 and corticosteroids. They all have receptors belonging to steroid receptor superfamily. And here you can see a generalized structure of the steroid hormone receptor wherein there is the N-terminal called as amino-terminal domain or N-terminal domain referred to as NTD or ABD. Then there is a middle which is the DNA binding domain referred to as DBD for the ease, and then there is a C-terminal which contains the ligand binding domain, that is the LBD. Although the names of these domains are explaining what they could probably be doing, but let us understand the role of each and every domain precisely. So, let us see the function of the first domain, which is the N-terminal domain. As you can see, it lies towards the N-terminal. This domain is actually highly variable in all the steroid hormone receptors. Here you can see that this NTD contains AF1, that is the trans activation function 1. Now this AF1 which is present in NTD is responsible for ligand independent activation. So some of the receptors, steroid hormone receptors, you will see that they can get activated even in the absence of ligand. So this AF1 is responsible for ligand independent activation. It recruits the co-regulators and core transcriptional machinery for the downstream transcription. It also modulates cell and promoter specificity of the target gene activation. Now, if we talk about the DNA binding domain which lies towards the middle part of this entire structure, this is highly conserved unlike the NTD, that is N-terminal domain. Now, this DNA binding domain contains two type 2 zinc finger motifs. So, there are two in number type 2 zinc finger motifs present in the DNA binding domain. Now, what is the function of this zinc finger motifs? They recognize and target the receptor to specific DNA sequences which are called as hormone response elements. So, please make a note of this thing that when the steroid hormone is there, this steroid hormone is already present either in the cytosol or in the nucleus. Now, what it has to do is whenever the hormone will come, it will bind to the receptor. Now, what is the function of this receptor? It has to go and directly bind to the DNA. So, keep in mind that the DNA sequence to which this receptor comes and binds is called as HRE, that is hormone response element. So, hormone response element is nothing but a DNA sequence to which the receptor will come and bind, and this is present, this basically is possible because of the zinc finger motifs which present in the DNA binding domain. After that, you can see in this structure there is a small H region, that is the hinge region. It is very small region but variable in all the steroid hormone receptors, and it contains NLS, that is nuclear localization signal sequence. So, if you could recall some of the cell and molecular biology lectures which you must have studied or listened somewhere, you are aware that NLS is responsible for intracellular trafficking and subcellular distribution of the proteins. Likewise, here also this NLS is present in the hinge region performing the same function. And then comes the ligand binding domain. So, please note that this ligand binding domain lies towards the C terminal, and this LBD region is highly conserved. So, it contains sequences which encode for heat shock protein association. So, you will shortly understand the role of these heat shock proteins. So, this receptor goes and binds to the heat shock protein unless it is active. So, this heat shock protein association is possible because of the sequences present in the ligand binding domain. Also, it is responsible for dimerization of proteins of the receptors and transcriptional activation and repression. What is important here is that it contains AF2. AF1 was there in N-terminal domain, and AF2, that is the activation function 2, it is present in the ligand binding domain. Now, what is the function of this AF2? It is responsible for ligand dependent activation. AF1 is responsible for ligand independent activation. So, whenever the ligand or hormone will come, this AF2 gets activated, and this will assist in robust transcription of the target genes by interacting with co-regulators along with the AF1. So, this is the last part of the structure, which is the C-terminal domain. This basically contains some retention signal, because of which the protein gets retained in the whatever the cell organelle wherein it has to be stored. So, for protein sorting, this terminal C-terminal domain plays a role. So, here you can see some of the gonadal steroid receptor structure. So, I have explained you the general structure. Now, you see in the androgen receptor, there are 919 amino acids starting from the N-terminal to C-terminal. Now, you would be able to understand the NTD, DBD, hinge and LBD domains. So, here AF1 lies in the N-terminal domain, AF2 lies in the LBD. Now, if you look at the estrogen receptors, there are basically two isoforms, estrogen receptor alpha and beta. Both of them are bit similar, but they do have some of the differences also. ER alpha is of 595 amino acids, while ER beta consists of 530 amino acids. What you can note here is that in estrogen receptors, these are also called as ESRs, they possess a C-terminal F domain. If you could see, there is additional domain called as F domain, which is not there in the androgen receptor. Also, this ER alpha and ER beta, they share 94% amino acid homology in the DNA binding domain. If you could recall, I have explained you that DNA binding domain is conserved. There is 55% homology in the ligand binding domain, and significant sequence divergence is observed in the N-terminal, hinge and C-terminal regions. Now, if you talk about the progesterone receptors, again there are two isoforms, progesterone receptor A and B. A consists of 769 amino acids, whereas the progesterone receptor B consists of 933 amino acids, and this is because of the additional N-terminal region which is called as B upstream segment, that is BUS. This consists of 164 amino acids, which contributes to the 933 amino acids in the progesterone receptor. So, I hope that the structure of the nuclear receptor are clear to you. So, the structure basically involved the N-terminal domain and the C-terminal domain, and in between there are hinge regions and DNA binding domain. So, now you are at a state of understanding that how these steroid hormones actually function. So now let us understand that. So number one, there is the genomic action, which is also called as classical model of steroid dependent and steroid receptor dependent method of action. Here what happens is that the steroid hormones come, they bind with the steroid receptors, which are present usually in cytoplasm or in some cases in the nucleus. So they directly bind to their receptor, which basically recruits the regulators and integrators for the transcription of the target gene along with the other transcription factors. So here in this figure you can see that how this steroid hormone, being lipid in nature or derived from cholesterol, it can very easily cross the plasma membrane, binds to the steroid receptor present in the cytosol, and can directly enter into the nucleus for performing the gene transcription of the target genes. So if you further see, let me explain you in a bit detail. So whenever there is diffusion of steroid hormone through the plasma membrane, it will go and bind to the receptor. Now you see that this receptor, which is labeled as steroid hormone receptor, SHR, here until the hormone does not come, it remains bind with the heat shock protein, HSP. I hope now you can recall, I have told you the function of HSP, which HSP association, which is performed by the activation factor 2. So here unless the receptor is activated by the presence of hormone, it remains inactive by getting associated with heat shock protein. So a complex is formed in the cytoplasm by the receptor and HSP when there is no hormone. Once the hormone comes, it will bind to its receptor. Now upon binding with the receptor, there will be conformational change, because of which this heat shock protein, which is shown by the green color, this will get dissociated, you see. So whenever the hormone will come, it will bind to its receptor, leading to conformational change, and then there will be dissociation of heat shock protein. Now what will happen next? This receptor will get phosphorylated and then it will move to the nucleus. So this small ball shown by the orange color is basically the phosphorylation, phosphate group attached to the receptor. So now it will come, and this you can see heat shock proteins are getting dissociated, and now this steroid hormone receptor gets dimerized. It is bound to its ligand and you can see that it is phosphorylated. Now once it gets dimerized, it will then bind to the HRE. I hope you remember what was HRE. HRE is the hormone response element, which is only the sequence of DNA to which the steroid hormone will come and bind. So you can see that how this hormone and receptor complex is getting bound to the DNA sequence, which is called as hormone response element in the target DNA. Once this binding takes place, now what will happen? It will now recruit number of coactivators and cointegrators like steroid receptor coactivator SRCP160, cointegrator CAMP response element binding protein, in short it is referred to as CBP, and other transcription factors which will give rise to the PIC, that is protein initiation complex. Now this complex, if you again I will say that if you study cell and molecular biology, you will get to know that this complex is the initial point from where the transcription will start. So now PIC will bind to the Tata box of promoter DNA, which will cause the initiation of transcription of the target gene, and thereafter the target protein gets formed. So this is one method, that is the genomic signaling, also called as classical method of action of the hormones, steroid hormones. Now recently the research has shown that these steroid hormones can also function via membrane bound receptors. So, this is called as non-genomic signaling, which is steroid dependent and steroid receptor dependent. So, receptor lies on the membrane. Now, once the hormone will come and bind to its receptor on the membrane, it will basically do crosstalk with a number of other proteins which will elicit the transcription. So, by this mechanism, either there is RAS, MAP kinase pathway gets followed, ERK pathway, PKA, PLC, all these are secondary messengers which gets activated once the membrane steroid hormone receptor binds with the hormone, and they will in turn give rise to the target proteins. Also, this mechanism can alter the membrane potential, and then there could be changes in the ion channels, and all these are non-genomic steroid hormone action mechanism. Now, if you compare both the methods, you will see that the non-genomic action is quite quick as compared to the genomic action. So, you will say that it is good that hormones must function very quickly. So, it is better that membrane receptors are there, so the non-genomic pathway is good, but it is actually not true. As you will learn through this series of lectures, you will realize that some of the hormones are specially meant to function slowly. Hence, the genomic action of the steroid hormones plays a significant role in our physiology. So, here I have kept one image wherein the entire pathway has been explained for the ease of understanding. So, this is how the steroid hormones act. Now, we will talk about the glycoprotein hormones, that is the GnRH, LH and FSH. Now, these hormones function via the membrane bound receptors, which belong to the largest family of membrane receptors called as GPCRs, that is G-protein coupled receptors. I am sure you must have studied about this receptor, its structure in cell wall biology, but let me take you to the brief introduction of the structure of GPCRs. So, this GPCR consists of an N-terminal domain which lies outside the cell, and then there are 7 transmembrane domains. So, that is why it is called as serpentine receptor as well in some books. So, there you can see here are 7 transmembrane alpha helices which are present in the lipid bilayer, and then there is the C-terminal domain which is present towards the cytosol and is also referred to as the intracellular domain. So, if you can categorize or divide this structure, there is the extracellular domain, there is a transmembrane domain, and then there is a intracellular domain of GPCRs. Now, what is important here is why they are called as GPCRs. If you see, G word stands for a G protein. So, G protein is actually a trimeric protein, consists of 3 subunits, alpha subunit, beta and gamma subunit. So, whenever the hormone comes, in general what happens is, whenever the hormone comes, binds to its receptor, it will activate this G protein, resulting in the segregation of G alpha subunit from the beta gamma subunit. So, here you see this G alpha subunit getting activated. Once this G alpha subunit gets activated, it will go and activate another enzyme which remains bound to the membrane. You can see here, which is referred to as adenylyl cyclase. So, it is present in the membrane. This is an enzyme which is membrane bound. So, this alpha subunit goes and stimulates adenylyl cyclase and it becomes activated. Now, what will it do? As the name suggests, cyclase, it will convert ATP into CAMP. Now, you must be aware that CAMP is the secondary messenger. So, what is the primary messenger here? So, primary messenger is the hormone. So, hormone comes and led to the activation of the secondary messenger CAMP. Now, this CAMP will amplify the reaction and it will now perform the downstream activation of a number of kinases, which will in turn cause the transcription of the target gene. Now, I must tell you that this G protein, G alpha protein is of different types like G stimulatory, G inhibitory, GQ. So, different types of G alpha subunit proteins are there. Now, in this figure, we must first know about the activation and inactivation of this G protein. So, here you can see that there is no hormone. So, under that scenario, what happens is that receptor remain bound to the G protein, and you can see that the alpha subunit of G protein is bound to GDP. That means it has only two phosphates associated with it. So, this is the inactive state. Now, whenever hormone comes and binds, this led to the transfer of one phosphate, and hence the GDP gets converted into GTP. So, whenever GTP is attached to the alpha subunit, it gives a signal that now the G protein has to get activated. Now, because of which what will happen, you can see here what I have explained you in the previous slide. Now, the G alpha subunit got activated, beta gamma also got activated. Now, they will target their downstream targets, like I have just explained you that G alpha subunit will activate the adenylyl cyclase. Likewise, beta gamma subunit will target the activation of phospholipase C, PLC. So, this once the G alpha protein gets activated, now there is a role of RGS protein, which is called as regulator of G protein signaling, which is responsible for GTP hydrolysis whenever the process or the function of the hormone gets completed. So, because of which you will see that again the inactive state is reached. So, after understanding the basic activation of G alpha subunit and G beta gamma subunit, now you are in a state of understanding that what happens when the CAMP gets formed by the action of adenylyl cyclase. So, once the adenylyl cyclase leads to the formation of CAMP, now the CAMP goes and binds to another kinase, which is called as protein kinase A. So, protein kinase A here you can see consists of two types of subunits. One is the regulatory subunit, which contains the docking sites for the CAMP, and then another type is the catalytic subunits. So, whenever CAMP comes, they will bind to the regulatory subunits, which causes the conformational change and thereby releases these catalytic subunits. You see, upon getting released, these catalytic subunits become activated and now they are entering into the nucleus. So, once they enter into the nucleus, what they will do? They will phosphorylate the transcription factor called as CREB. You can see what it is doing. It is doing, see, kinases, the role of kinase enzyme is definitely to phosphorylate another protein. So, what protein kinase A does, it phosphorylates the CREB transcription factor. You must know the full form of CREB also. So, CREB stands for CRE binding protein. B stands for binding protein, while CRE is the CAMP, C stands for CAMP, RE is the response element. So, CAMP response element binding protein is what is CREB. So, once this CREB gets activated, it will now recruit the coactivators and will now perform the transcription of CAMP inducible genes. So, after understanding the basics of GnRH, LH, FSH hormone receptor structure and how do they perform their action, now let us move towards more specific concept, that is the GnRH. So, let me tell you that GnRH is a decapeptide hormone. It just consists of 10 amino acids which are shown here, out of which the 3 amino acids arginine, proline and glycine are responsible for binding to the receptor, whereas the remaining on the other hand end of the this glycoprotein hormone, these 3 amino acids helps in the receptor binding and activation. So, this GnRH was first discovered from the pig hypothalamus by the rigorous work by Shelley in 1971. Now, this GnRH is produced actively in the GnRH cell bodies which are present in the hypothalamic areas referred to as MPOA, that is medial preoptic area, and arcuate nucleus. So these regions of hypothalamus contains the neurons which actively synthesize GnRH. And what is important here is that GnRH does not constitutively gets expressed. It is released in the pulsatile manner. So GnRH functions via its receptors which are definitely present on the anterior pituitary. I hope you remember the endocrinology series wherein I have explained to you that how GnRH goes and stimulates the production of the gonadotropins. So gonadotropins, FSH, LH has to be produced from the anterior pituitary, hence the receptors of the GnRH must lie in the anterior part of the pituitary. So the GnRH will be released from the cell bodies in the hypothalamus and then they will bind to the receptor present on the anterior pituitary. Now these GnRH receptors are GQ linked GPCRs. I have explained to you that G alpha is of different types like G alpha, S, G alpha, I, G alpha, Q. So GnRH is GQ linked GPCR, and what is interesting here is that the C terminal is quite short in length. Now, how it is important that this short intracellular C terminal domain of GnRH makes it different from other GPCRs, because if the tail is short, it will slows its internalization, and hence if it is not getting internalized, it will not get desensitized. So it prevents rapid desensitization of GnRH receptors. So most of the GnRH receptors are GQ linked, but it is worthy to mention that some of the receptors are also reported to be GS protein coupled, which stimulates CMP PK signaling pathway, which we have just studied few slides back. So once GnRH gets synthesized, it will definitely go and leads to the production of the gonadotropins. Now you must be wondering that how GnRH, just one hormone, can control the production of both LH and FSH. So very interestingly, whenever the GnRH is produced in the higher rate, that is rapid pulses of GnRH are produced, this leads to the LH formation and secretion. Whereas if the GnRH pulses are slow, this leads to the production and secretion of FSH. So in this figure you can see that how GnRH, once the GnRH comes, binds to its receptor, it causes the activation of GQ protein. Once this GQ protein gets activated, it will cause the activation of again another enzyme which is membrane bound called as phospholipase C. Now once phospholipase C gets activated, what it does, it goes and breaks the PIP2 into IP3 and DAG. Now DAG is diacylglycerol and IP3 is the inositol triphosphate. So both of these DAG and IP3, just like CAMP, both of them are the secondary messengers. Now this DAG will cause the activation of protein kinase C, which then causes phosphorylation of T snares, V snares, which help in the exocytosis of the LH and FSH. So these colored balls are actually representing the LH and FSH. These dark colored balls are the FSH, which are more in number. The lesser in number are the LH hormone, which is getting exocytosed by the action of phosphorylation of V snares and T snares by PKC. Now if you look at IP3, IP3 goes and binds to its receptor present on the endoplasmic reticulum, which then causes the exocytosis, intracellular calcium release from the ER. Now this intracellular calcium release regulates the exocytosis of LH and FSH. So this is about the GnRH signaling. So in today's lecture, what we have studied that the hormones which are important to the reproduction could be classified into steroid hormones and in the glycoprotein. Now the steroid hormones function via nuclear receptors, but recent research has shown that there are membrane receptors. So, we have studied the structure of these steroid hormone receptors which are in the nucleus or cytoplasm. After that, we have studied that how these steroid hormones can function via genomic pathway, which is the classical pathway. For a very long time, we all believed that hormones, the steroid hormones function via these nuclear or cytoplasmic receptors. But then we have seen that there is another non-genomic pathway, which is not classical pathway of steroid hormones mechanism of action. After understanding the steroid hormone mechanism of action, we moved to the glycoprotein hormones, and we have seen that how these GPCRs, what is their structure and how GnRH functions. Now, in the next lecture, we will understand about the mechanism of action of FSH and LH and some other hormones that play significant function in reproduction. Thank you.